So I'm going to be talking about one of the components of the land-ocean continuum, estuaries, looking at carbon transformations and particularly CO2 exchanges in estuaries. So you might wonder, why do we need some more estimates of CO2 exchanges? There are a million out there already, from the deep eutrophied estuaries in Europe to the nice shallow ones in Australia to the northeast, the US East Coast. So why do we need more? What's often, sorry about this, what's often been missing in estimates of CO2 transformations are the mechanisms responsible. So I'm going to drill down to try to understand what are the major drivers in estuaries that regulate CO2 exchanges. So one of them you'd expect would be net ecosystem metabolism. And so this isn't where here we go. So freshwater discharge delivers lots of stuff in the form of dissolved, in particular organic carbon, dissolved inorganic carbon, nutrients into the estuary, where the nutrients support gross primary production, which produces DOC, both as photosynthate or due to grazing, and then the DOC is respired to form DIC, and the DIC then can be partitioned. Some of it goes into the atmosphere, some to the ocean. But this isn't the only DOC that is respired in the estuary. There's all the DOC coming in from the allochthonous DOC. Similarly, DIC, DIC can be supplied from rivers, especially those that are bordered by marshes, which we'll discuss in a bit, also release a lot of DIC. So we'll be looking at how NEM affects CO2, fluxes, and then so in general, what are the drivers of CO2 fluxes? Hydrology, I'll talk about freshwater discharge and freshwater age. Temperature, of course, I've mentioned allochthonous inputs, but it depends on where they're coming from. Are they coming from marshes, industry, or urban sources? Forested systems, which release a lot of humics, and groundwater, which often has a lot of DIC in it. And autochthonous inputs I've also mentioned. And finally, alkalinity can be delivered in rivers, but it's also produced in the estuaries by nitrate and sulfate reduction. And in eutrophied estuaries, that could be an important source of alkalinity. So in this, I'm going to first start off talking about my own research in two different systems, the York River Estuary in Virginia and the New River Estuary in North Carolina. We asked 4 questions: how do air-sea CO2 exchanges vary in these systems during years with very different precipitation patterns? Secondly, how does freshwater age influence NEM and air-sea exchange? And I'll try to identify what are the direct versus the indirect regulators of CO2 exchanges. And finally, I'll compare our estimates to those that have been made along the east coast of the US and also that have been modeled. So here are our systems. The New River Estuary is just south of where the Outer Banks end. And we split the system into 7 boxes. And I'll be giving estimates per box. Here's the York River Estuary in the western shore of Chesapeake Bay. We've split that into 5 boxes. And these are our cruise paths, zigzag path, which covers about equal amounts of shoal versus channel. So a comparison of the systems, and please just look at the shaded portions. The York River Estuary is much bigger than the New River Estuary. The mean discharge in the York is an order of magnitude greater than the New River. However, interesting, flushing time is similar. So that's because the New River Estuary is a totally weird estuary. It's a series of linked lagoons with a barrier island at its inlet, which sort of slows down discharge. So export. So flushing time ends up similar. And the New River Estuary has a city at its head, city of Jacksonville, whereas the York River Estuary is pretty much rural, no cities. So just to show how the freshwater discharge or flushing time varied in our years, study years compared to the long-term annual averages. So the blue bar represents the 20 or so year average for the York River system. And this is the year we made our measurements. Freshwater discharge was about twofold higher than the long-term average annual discharge. And in the New River Estuary, we made measurements during two very different years. 13-14 was a drought year. Freshwater discharge was lower than the long-term average, and in 2014-15, it was higher. And then flushing time varies also with discharge. So in the York River Estuary, flushing time was very short compared to the long-term average, whereas in the New River Estuary, during a drought year, it's about average, and 14-15, it was slightly above average. Flushing time makes a big difference to things like production. If flushing time is really short, the phytoplankton don't have time to double and they don't accumulate. And so, sorry for all these words. I tell my students never to have slides with all these words. However, I think it's really important that I explain how we measure net ecosystem metabolism, because it's quite different than the way most people do it. We use the open water method. I make bi-monthly in each system. We run what's called a data flow cruise, starting at dawn, like 4:30 this last week. So at dawn, at the following dusk, and the next morning at dawn. Okay, and we did that for all of the systems. And in those data flow cruises, we pump water from about a half-meter depth. The water is split. Part goes through a YSI, a CDOM sensor, the other through a shower head equilibrator to an infrared gas analyzer. And for these NEM measurements, we took DO data. We distance weighted it in each of the boxes, and then interpolated over 24 hours. Then we determined gas exchanges using pretty much standard methods in the model of Jean Gadal for the gas transfer coefficient. And then we calculated daily NEM using average depth for each box and corrected for air-sea exchange. So that's the NEM data that I'll be presenting. And so some of our results. So CO2 fluxes. Whoops, here we go. So CO2 fluxes in the York River, I'll start out with February and March. It's cold, there was a spring bloom, and there was very little, discharge was really low. And so there were no CO2 fluxes out. There was a little bit of uptake. And then it started to rain in May, and it's still raining. It rained through the fall, and the whole system became a source of CO2 throughout the estuary. Now, the New River Estuary is a little bit different. The only place we saw, this was a drought year. So the only place we saw CO2 fluxes were at the very head of the estuary. That could be a pH issue. There was more CO2 to flux out. But for the rest of the system, going down the estuary, there were either no fluxes or there was uptake. And then in a slightly wetter year, 14-15, so again, fluxes at the head of the estuary, but in spring and fall, there were fluxes throughout the whole system. On the other hand, well, excuse me, during other months, there were basically no net fluxes out. So the only time we saw export was in spring and summer, spring and fall. So now how about NEM? That was the other thing we were keen on measuring. So in the York River so everything above the x-axis represents autotrophy, everything below heterotrophy. So when the system is cold and there's low discharge, like February and March, and there was a spring bloom, it was net autotrophic. the rest of the year, it was basically net heterotrophic, except for one point. We're not sure of that one, especially since chlorophyll was low. And then in the New River estuary, during a drought year, there were no patterns in net ecosystem metabolism. It was all over the place. But in a wetter year, 2014-15, you can see that the New River estuary, for the most part, is net autotrophic, quite different than the York River. That may be because of all those pigs in the watershed and a lot of agriculture, a lot of nutrients coming into that system compared to the York River. So now, freshwater age we had observed before that freshwater age plays an important role in fluxes. So in the York River estuary, below about 20 days, now that means that parcel of water has been in the estuary for 20 days. So that's water that's just arrived and sat around for 20 days. During that period of time, a lot of CO2 fluxes out of the York River estuary. And then after 20 days, nothing. In the New River estuary, oops, I seem to have trouble with this. In the New River estuary, my point disappears. Below about 10 days, there were CO2 fluxes that were small, and after that, there was nothing. And then in a wetter year, again, in the New River estuary, there were fluxes out at a freshwater age less than, I'm going to use the pole, less than 20 days. And so finally, nettrophic status, how does that vary? So again, everything above the x-axis is net autotrophic. So below 20 days in the York River, it was net heterotrophic. Then it switched to net autotrophy. Whereas for the New River estuary, in both cases, NEM was really weakly related to freshwater age, although there was a pattern of net autotrophy at short freshwater ages. So it was exact opposite pattern as the York River. Here, net autotrophy, here, net heterotrophy. Again, the New River estuary is a weird system. And finally, just to remind you all, effluxes of CO2 occur in net heterotrophic systems. CO2 uptake occurs in net autotrophic systems. And there are also other drivers. For example, DOC and DIN were strongly related to CO2 fluxes. They were always highest at the head of the estuary, decreased down estuary. But interestingly, chlorophyll was weakly related to NEM and not related at all to CO2 fluxes, although you'd expect it would be, but it wasn't. So we then did a structural equation model to try to get at what are the direct drivers versus the indirect drivers of CO2 fluxes. And so in this model, if it's a solid line, either black or red line, it means that it's significant. Whereas if it's a gray line, those are all insignificant. If it's red, so for example, freshwater age is related inversely to DOC. Freshwater age goes up, DOC goes down. But it was a direct driver. DOC is a direct driver of DIN, and it's a positive relationship. And you'd expect that if DOC is being remineralized to form that DIN. And both DIN and DOC are direct drivers of CO2 fluxes. Interestingly, NEM is also a driver, but there was a much weaker relationship, probably because these are annualized numbers, and so there's a lot of variation annually. As you'd expect, temperature is inversely related to NEM because there's greater support of respiration relative to photosynthesis. And notice chlorophyll, everything is insignificant for chlorophyll. So then the next question we asked, okay, if there are all these transformations of carbon in the estuary, where is the carbon coming from? So one possibility is the DOC and DIC are from riverine marshes. So Pete Raymond published in 2000, pointed out that estuary and DIC measured in the system was in excess of carbon fixed plus DOC respired. So it had to have been coming from outside of the system. And then at about the same time, Scott Neubauer and I determined that riverine marshes could supply about 47% of the excess DIC that Pete was measuring in the estuary, although some of this was going off into the atmosphere as CO2. However, we didn't observe any DOC, or it was negligible DOC export from marshes. Now, Bryce Van Dam, for the New River estuary, using the equations of Joseph et al., observed that in the New River estuary, new riverine input was not as important as internal transformations. So quite different from the York River, partly because of that long time freshwater, not discharge, the other one, whose word I can't think of right now. And so just to show that marshes can release a substantial amount of DIC, not necessarily DOC, I've just included, these are all pretty substantial numbers. I think Wei-Jun showed lots of DIC export from the Georgia rivers, but not that much DOC export. Similarly, Knobloch et al.'s student at VIMS has been showing, again, DOC export, there is a little bit of DOC export, but it's very variable depending on tides, and there's a lot more DIC export from the Tascanis Creek. So then I thought it would be fun. Pete Raymond did a measurement in the York River estuary 1996 and 7, and we did it in 2018. We used different methods. So how close are our measurements? Good question. So, so both Pete and we observed highest PCO2 in summer and fall. However, Pete said that was because residence time was longest, and we observed that residence time was shortest. We also observed the lowest PCO2 in winter and spring when there were low temperatures, spring bloom. Pete said high discharge. We said low discharge. So on the other hand, when you actually look at the measurements, for example, NEM, and we measured them totally differently, they're basically exactly the same. And fluxes of CO2, ours were a little higher, but we had higher discharge. So different methods, slightly different interpretation. Net heterotrophy is a main driver. In the York River, it was very supersaturated, but we believe freshwater age really plays an important role in determining fluxes. So then I compared, why does it say 6 minutes? I mean, 6 minutes left, I guess. We compared our estimates of CO2 fluxes to those made along the whole east coast of the US. I've ordered these north to south. So we'll start in New Hampshire. These estuaries, the Cocheco is highly eutrophied with 2 wastewater treatment plants. All these systems have short residence times, very variable discharge, and very low fluxes of CO2 based on Hunt. Then if we move south to Delaware Bay, a very big system, Joseph et al. Split the Delaware Bay into 2 parts. The upper bay is highly eutrophied because of things like the city of Philadelphia, and so it was net heterotrophic. The lower bay is much more surrounded by more rural areas. There's a lot of mixing. It was net autotrophic. So if you need one number for releases of CO2 from Delaware estuary, it's fairly low because the upper bay balances the lower bay. In the York River estuary, well, I've already mentioned very high discharge, pretty high fluxes of CO2. Pete, similar numbers, lots of heterotrophy, lots of allochthonous inputs into the York River. The New River estuary have 3 different studies done slightly 3 different times. 13, 14, a period of low discharge, low CO2 fluxes. And then between 2014, 16, 2 different studies. And the fresh fluxes were pretty similar, and they were about sort of middling freshwater discharge. It was slightly above average. And then the Neuse River in North Carolina is close to the New River, so it's only miles away, but it's a really different system. It's a much bigger river. It ends in Pamlico Sound. It has flushing time is very slow. Riverine input is really important. So a lochliness input is supplying the carbon to the Neuse River. However, discharge overall, well, maybe it's not that different from the new. It was fairly low, maybe because of that long flushing time. Then we get to Georgia, where there's fluxes are a lot higher. So the Altamaha is a big, big river, huge watershed, high discharge, high fluxes. And the other rivers, I think the Satilla goes as far as, they're all coastal plain rivers. I think they also have high fluxes, but they're also impacted by very high inputs of dissolved inorganic carbon from the very productive marshes in Georgia. So then, Laruelle, Galvan, modeled, and I read the paper and say, why are we making all these measurements when Galvan has a model and he can predict all these things much easier than, ours are very labor intensive. So I thought, let's compare his mid-Atlantic measurements right here, 11.1 for FCO2, to what we measured in the York River, mid-Atlantic estuary. And they're not that far off. Now, Galvan's model, he can explain it later, is based on morphology. Funnel-shaped estuaries are different than our lagoonal shaped like New River estuary. So if you compare the fluxes from his model to the New River, you see they don't compare that well. Now, fluxes of NEM, he calculated it for mid-Atlantic, and it's very similar to what we calculated for the York River, but not so much for the New River. So it does a good job for lots of estuaries, but there are some that don't quite fit the model. And then Galvan took all of the data from the East Coast marshes and he developed a regression. So we used his regression coefficient to calculate CO2 fluxes based on NEM. And again, it's good for the York River, pretty good, not so good for the New River. So it does a pretty good job. And certainly, that's really, you couldn't possibly do the labor-intensive measurements we do in lots of estuaries. So you have to be able to model it. So what are our take-home messages? Freshwater discharge plays an important role. It's delivering nutrients and also DIC and maybe DOC. And it is a major driver. It has to be of NEM. And NEM, if it's net heterotrophic or autotrophic, determines the CO2 flux. We believe interannual variability that you see in a lot of systems is due mainly, or a lot of it, to freshwater discharge. Temperature also plays a role. I should mention the biggest difficulty in estimating these fluxes is what do you do with extreme events? North Carolina has a lot of hurricanes. And those extreme events, one event, Bryce Van Dam observed that one event, Hurricane Joaquin, there was as much CO2 flux in that one event as for the rest of the year. So it's not clear how you take that into account in models. Also, spatial variability within a system that's usually ignored is, we believe, determined a lot of it by freshwater age. And then we believe that if you have rivers bordered by lots of marshes, they are important sources of DIC. That's what the lateral flux people are trying to figure out. Where is all that DIC? Where is the carbon coming from to produce all that DIC? DOC, there's a big question about the importance of marsh sources of DOC. And finally, we recognize how difficult it is to simulate reality in models, but I really appreciate them. Thank you.
Thank you, Iris. So questions for Iris?
Hi.
Go ahead. Do we have one question?
Hi, Jim Bishop, UC Berkeley. I've worked with the Eel River Critical Zone Observatory, and in that location, we were following the water flowing from groundwater into the Elder Creek, feeding the Eel River. And it seems that a lot of the DIC production is due to weathering reactions expressed in groundwater. And I'm wondering how your estuarine systems express runoff, surface runoff versus groundwater inputs for DIC.
So we measured groundwater inputs in, sorry, here I can use this.
Yeah.
We measured groundwater inputs in the New River and they were negligible. Okay? There are no estimates for the York River that we could use. I have a student now that's looking for discharge from this. There is a subterranean estuary at the mouth of the York, and she's in the process of trying to calculate whether that discharge is negligible relative to what's coming down the river. I believe Wagens also, you've also measured, taken into account groundwater discharge.
Yeah, I believe it's relatively small.
Yeah.
Yeah.
Jane Caffrey did a lot of those California. She used NERS for the National Estuarine Research Reserve. There is a lot of data, but those are all shallow systems, and a lot of them were net heterotrophic.
We have a question here.
Iris, it's really nice to see you put the East Coast estuaries together. But one obvious end member would be Chesapeake Bay,
That's true.
So look at the spectrum of water residence time. What do you guess for the Chesapeake Bay? It's,
The residence time in the bay?
No, you put this spectrum of different residence
Where
time,
it
and
would
you see,
be?
like, for example, in Delaware Bay, head of the upstream, there's a large CO2 source, but downstream it's very small. So average it become a lot smaller. But the Chesapeake Bay is even, goes to that end member with a very, very long water residence time stratified. Would you guess it's a CO2 source or a sink? And metabolism.
Well, in a sense, it's like, and you're doing this right now, right? So the upper part of the bay is usually net heterotrophic, and the lower bay is net autotrophic. And so they're, in a sense, balancing each other out. And you have all that stratification in summer, which can be very, very extensive, which is where you get a lot of DIC that accumulates in that below the picnic line, and then is that mix. I'm trying to figure it out.
Actually, I was trying to support you because our observation shows wet year or dry year make big difference to change that system. So
Well,
I think you
for
are
the same reason,
yeah,
right.
we're on the same page with that.
So when you mix in the fall and winter, all that DIC comes up and then moves down. Some of that is being released in CO2. So is that substantial? Compared to
October, you do see late October when the mixing starts, you see CO2 release. But winter time, it's very low.
So overall, it's probably like
Yeah.
Delaware Bay, with the whole thing balances out. And you don't
It could
get
be
on
a
an
sink
annual-
in some sense because it's stratified. Delaware
Yeah,
Bay is
yeah.
not stratified.
Yeah, right.
Okay, I think we've got one more question, another question in the back here.
Hi, Nick Nadziko from Santa Barbara. Maybe this will happen in the panel instead. I was just curious about how you calculated freshwater age.
Freshwater age, they use, Mark Brush did that actually. I left out the thank you acknowledgement slide. He used the box model of officer and used salinity to calculate and freshwater discharge to calculate. Yeah, but no, then he. No matter what I say, Greg always contradicts.
Okay,
Been doing that for a long time.
I guess we better move on to the next speaker. Thank you very much, Iris.
Before.
Okay, thank Iris again.
I have.